I'm going to talk about the use of TMS in language research, and I realise that some people will be familiar with TMS and some won't, but I'm still going to give a background because it's the first in the series of talks. I thought that might be helpful. So TMS is a relatively new technique in comparison with other techniques used in cognitive neuroscience, and it was first used in humans to stimulate motor cortex by Barker et al. 1985 in Sheffield University. And since then it's been applied to answer a whole range of questions in cognitive neuroscience, including language. So just an outline of what I'm going to go through today. First of all, we'll just have a general look at the TMS method and the advantages of the TMS method, and finally an outline of some of the examples of the different ways to use TMS in language research. So the examples I use are just kind of one example to represent a type of use, but it's not to say that these are the only studies that have used TMS in this way. So the principle of the TMS technique then relies on electromagnetic induction. So basically when you've got a current running through a coil, it induces a magnetic field, and if that magnetic field is changing, it can induce a current in a nearby conductive material. So in TMS then, the current running through the coil provides the setup to induce a magnetic field, and this magnetic field is changing and induces a current in the underlying cortex. So this is the standard TMS machine, which many of you will be familiar with, the standard figure of 8 coil, which will be used in all of the studies I'll be discussing today. Okay, so basically then, the technique relies on a peak in a current in the coil, which happens at around 0.1 or 0.2 of a millisecond. And this results in a peaking magnetic field. And at the points when the magnetic field is changing most is when you get the greatest amount of induced electric field in D. And that induces current then in the cortex, as you can see in E. And all of this happens within a very short period, in the first 0.1, 0.2 milliseconds. And at the bottom then you can see that the behavioural effect usually seen, at least for high-frequency TMS, is an increase in reaction time when you're trying to do a task for which the area of cortex that you're stimulating is necessary. Okay, so how are the neurons activated? So each of these cylinders represents an axon. So in A we can see that the axon is lying in a field that's running parallel to the axon, so no current is induced, whereas in B there's a change in the electric field, so at some places it's larger than others, and this causes hyperpolarization and dehyperpolarization at other points, causing a current to run through the axon. Another way where a neuron can be activated is if there's a bend or branching in a neuron, which we can see in C. So this means that the magnetic field experienced by the axon is not uniform along its length. And again you have hyperpolarization and depolarization, which causes a current to run through the axon. Okay, so the distribution of the induced electric field depends on the type of coil you use, and all the studies I discussed today will use a figure of 8 coil on the right rather than a circular coil. But you can see that the maximum induced electric field is marked in red here in the figure. So on the left you can see that in a figure of 8 coil it's a much larger area that is affected, whereas on the right the figure of 8 coil gives a far more focal region. But even though you get a focal area, there's still a spread of the magnetic field. So in order to refine the inferences you make from your results, you can basically move your coil to different positions, to neighbouring positions on the scalp, in order to refine the region you think that's important for a particular task. Okay, so in comparison with other techniques used in cognitive neuroscience, spatially, the spatial resolution of TMS is actually quite good. So it's around, well, some people claim less than a centimetre in terms of millimetres, but I would say a centimetre at least. In temporal resolution then, it's also got very high temporal resolution, so far better than techniques such as fMRI or PET. And also TMS is an interference technique rather than a correlation technique. And this is extremely important when you're looking at areas of cortex that are actually necessary for a process rather than just activated unnecessarily. Okay, so two factors pretty much decide whether you'll get a TMS effect during processing. So on the top left you can see again the peak of the intensity of TMS in the first 0.1 or 0.2 milliseconds. And the intensity and the probability that a region is involved in a task is what decides whether you get a behavioural effect. So in the larger curve, the blue curve that shows probability of an area being involved across time, you can see that at point A, you're unlikely to get an effect because the intensity of TMS is low, but it's also it's not very probable that the region is involved in the task either. Whereas later on, if you've got relatively high probability of the area being involved and you've got a reasonably high intensity of TMS, then you are quite likely to get an effect. And this also shows that you can get an effect at different time points along the time window when an area is involved in a task. And this explains why the peak of an ERP doesn't always correspond with the times you can get an effect of TMS. Okay, so for localizing the sites for stimulation, most people nowadays use frameless stereotaxic setups. So for this you need a structural MRI scan of an individual, an infrared camera, and tools that can reflect infrared light. So you can see the subject sitting here with a tool that has light-sensitive balls on it and also a marker on the head. So it shows the position of the head. And if you register the position of the head in space with your MRI scan by choosing locations such as tip of the nose or the ears, etc., then you know exactly where in space the head is. Then you can select a site by marking the region you want to stimulate on your MRI scan, and then putting your coil on the scalp with the sensor attached. You can show exactly where on the scan you will stimulate. If you don't have an MRI scan for everybody, which in our study was the case, so half of our subjects didn't have scans, you can basically get average scalp measurements from people who do have scans. So we asked 10 people to come in, we marked the sites we were going to stimulate, and then we got average scalp sites. And it's actually surprisingly accurate once you get a big enough sample of people to find an average scalp measurement for the site required. And then on the right, a far less kind of useful way of doing it, which people used before frameless stereotaxic systems were available, was to mark the vertex of the head and from there find the hand cortex, which of course you can stimulate to know that you're in the correct region. So you stimulate the hand cortex and see movement in the hand. And from there you basically try to work out the distance you need to be at the site you want to stimulate. So here roughly inferior frontal cortex. Okay, so there are obviously several advantages then to the technique. First of all, it's non-invasive, which is necessary for use with normal humans. Secondly, it allows us to interfere with activity, which is a novel thing for a technique in neuroscience. Thirdly, it allows us to imply causality. A lot of techniques, such as fMRI, are correlational in nature, so we don't always know that activity we see in the brain is actually necessary for the process. And finally, then, the setup is actually relatively inexpensive in comparison with other methods. So there are disadvantages too. First of all, there can be discomfort when you stimulate. So if you're stimulating frontal cortex, as we were, then you can get associated muscle movement, which can be uncomfortable. But subjects usually muscle twitches. So that can be quite uncomfortable for subjects, but they usually get used to it. And also, of course, there are restrictions on use, and you can't use TMS on children, for example. Okay, so the original focus then on language research was on the speech element, probably because it was such an easy effect to see that you could stimulate posterior, like motor cortex or posterior Broca's area, and prevent people from continued flow of speech. And that was quite attractive because you could see the effect so easily, the behavioural effect. Later on, so Pascaloni did this first in 91 with frequency of 25 hertz, but later on Epstein showed that you could do it like at 4 to 8 hertz, so much lower frequency. And then Stewart et al. In 2001 also showed speech arrest, but this time they tried to move beyond just motor effects. So they showed that a more anterior site from the motor cortex could give speech arrest without motor evoked potentials from the face. So basically a non-motor effect, but still affecting speech. Okay, so later studies then moved on to look at more cognitive levels of language processing, and that's what we're going to be looking at. Okay, so these are basically the three broad uses I'm going to discuss. The first type is 1 Hertz TMS, i.e. a pulse every second. And you can do this offline, i.e. when a subject is not actually performing a task. So a subject performs a task, then you can apply TMS for, say, 10 minutes, and then they do a task afterwards, so you have a pre and post comparison. And a good example of this is Naser et al. 2005, and I'll look at that study in a moment. I realise, having spoken to you, that a lot of you are actually familiar with this study, but I'll still go through it. The second type then is another type of repetitive TMS, but it's much higher frequency, so 10 Hertz TMS. And that's online, i.e. when the subject is actually performing a task that you apply TMS on particular trials. And I'll discuss the study we did at Oxford for that. And then the final type is single pulse, so that just refines the time. So in the 10 Hertz example, you'll see that you can find a time window when a region of cortex is necessary, but that can be up to 300 to 500 milliseconds. And if you want to refine it beyond that, you can use single pulses to go in at much shorter times. Okay, so the first study then, by Naser et al. 2005, is a 1 Hertz RTMS study, as I said, offline. And this was with aphasic patients, looking at long-term effects of TMS. And this study is basically based on an earlier study done by the same group in 2002, where they found a benefit of RTMS in the right homologue of Broca's area in pars triangularis. And they found that after 10 minutes of 1 Hertz TMS, people had improved naming. And this study was a follow-on to see that if you increased the amount of TMS that an individual had, would the effects be longer term? Okay, so there were 4 patients involved, all right-handed, 5 to 11 years post-middle cerebral artery stroke. The first patient was recovered from non-fluent aphasia. 2 and 3 were mild non-fluent aphasics, and the 4th had severe non-fluent global aphasia. Okay, so this is just to show their lesions. And the first 3 are actually similar, and they've got damage to kind of traditional Broca's area, whereas the final one, the damage seems to be mainly subcortical.
Excuse me, the 4th patient is the one with the most severe aphasia, that was the
yeah.
global.
Yeah. Okay. Right, so as I say, the stimulation was in right hemisphere pars triangularis, that's Brodmann's area 45. They had 1 Hertz TMS for 20 minutes on each day, And the threshold was 90% of motor threshold, so sub the necessary activation required to cause a motor evoked potential. And they had a TMS session for 5 days a week for 2 weeks, so that's basically 10 sessions overall. Okay, so this is just a picture of their experimental setup. You can see they use a figure of 8 coil, and also that they use the online stereotaxic system. And just to point out, the frameless stereotaxic system, if you're stimulating for a long period of time, is actually a very good idea to use it online, because any movement of the coil whatsoever can change the area of the cortex that you're actually stimulating. Okay, so the behavioural measures were the Snodgrass and Van der Vaart picture naming, pre and post RTMS, the Boston naming test, and this was measured pre-TMS, 2 weeks post, 2 months post, and 8 months post. And then finally the Boston Diagnostic Aphasia exam, and 2 subtests of that, the animals category and tools category. And they were also tested at pre-TMS, 2 weeks, 2 months, and 8 months post. Okay, so these are the overall results for the group then, for the first test. On the left you can see that the number of correctly named pictures increases. So the black bars represent TMS treatments, and the white bar is pre-treatment. So for all patients there was an increase in the number of pictures they could name after TMS. On the right you can see that the reaction times also improved for the first 3 patients, but not the 4th patient, well, not significantly at least. Okay, this is just a table then of the results for the other 3 tests. The first column then is the measure for the pre-TMS result, and the 3 columns after then are 2 weeks, 2 months, and 8 months post. So you can see for the Boston naming test, there's an improvement across the group for 2 months post and almost 8 months post, but not quite significant. And then for the other 2, for the Boston Diagnostic Aphasia exam and the animal category, there's an improvement 2 weeks, 2 months, and again almost 8 months post-RTMS. And then the final test, only the 2-month and 8-month post periods showed an advantage over the pre-TMS measure. And these are graphs of the individual subjects for each of these tests. So you can see there's a fair amount of variation between the subjects. So subject 3 looks to do fairly badly all the time. 1 and 2 look to improve pretty much on all of those. But there's actually, yeah, a fair amount of variation across the group. But over the group in general, the mean shows an improvement. Okay, so just to summarise that then, there's improved reaction time and accuracy after 10 sessions of RTMS treatment of pars triangularis. The study was also set up to look at the safety of using TMS like this, so over such a long period, whether it would be safe to do this, and apparently it is. There were no side effects. But importantly, and the authors point this out themselves, they need a control condition. So great, they got an effect after right hemisphere stimulation, but there was no control site. So there's nothing to say that the effects aren't due to something else. And also, I think it needs fMRI data to explain the differences. So we know that, yes, if you stimulate right hemisphere, you get an improvement, but what does that actually mean? What's the change in activations? So you need a pre and post fMRI test to see what the changes in activations are.
And did they do any kind of error analysis about, in terms of what kinds of errors these patients made initially, and then what kinds of errors dropped out or improved?
Yeah, I'm afraid I don't know.
Error pattern looked like what you see in recovery, or was it something else?
I don't actually know.
They didn't do an item analysis or error analysis.
Right.
So they didn't segregate items into semantic categories or any any variable that you find.
Which is too bad, because I'm sure they have all their data.
Yeah.
Okay, so then the second type of TMS I'm going to look at is a much higher frequency TMS that looks at much shorter effects in reaction time. So this is 10 Hertz RTMS, so a pulse every 100 milliseconds and 3 pulses in every train. This is online, so while the subject is performing the task. We used a standard 60% machine output rather than measuring the motor threshold. And certainly there could be some differences in motor threshold, but we used a standard 60%, which is, I would say, above the motor threshold for most people. So again, as I say, we're looking at short-term effects on reaction time. But the study was looking at whether or not you can dissociate processing of semantics and phonology within Broca's area. So previous imaging studies saw that there was a possibility that anterior Broca's area seemed to be more involved in semantic processing, whereas posterior area seemed more involved in phonological processing. And we wanted to see whether if you stimulated posterior areas of Broca's area that you would get an increase in reaction time for phonological task but not semantic, and in anterior Broca's area whether you'd see an effect on semantics but not phonology. So we're looking for a double dissociation. So our definition then of left inferior frontal gyrus is Brodmann's areas 6, 44, 45 and 47. So I've shown Brodmann's map on the left. And then on the right, pars triangularis, pars opercularis and pars orbitalis. We were aiming to stimulate in pars opercularis as our posterior site, and then on the border of pars triangularis and pars orbitalis as our anterior semantic site. Okay, so to localize the sites, we first did this anatomically by using the frame stereotactic system. And then after that we did a functional localizer. So in the semantic session, we would ask subjects to do a judgment on a pair of words to decide whether the pair of words belong to the same category or not. And then we would stimulate the anterior site to see if we could see an increase in reaction time while doing this task. And if we saw an increase in reaction time on 2 blocks of this task, then we would say we had found the correct site that we were going to use for the main task. And if not, we would move 1 centimetre away and continue until we found a site where there was an effect on 2 blocks. So also for the phonological localizer, we did the same thing, except this time subjects were deciding whether a pair of words rhymed or not. And again, yeah.
Sorry, do you mind if we interrupt with questions?
That's fine.
So when you say you moved a centimetre away, in what direction? And how many times on average did you move a centimetre away in order to find the optimal site?
So the posterior site was probably much quicker to find usually. And we probably only had to move twice, 3 times maybe. The anterior site was usually more difficult to find. For the first site we would usually move posterior from the original site. And the anterior site we would move anterior and ventrally. So we were basically moving along the border between 45 and 47 or into 47. But I'll show you where we ended up anyway after moving around.
I'm sorry, Patricia, I'm confused about just the timing, if you wouldn't mind just walking through that again.
Okay.
So it's, what you said is that this is 10 Hertz and you said 3 pulses per train.
Yeah.
And so how many trains does the subject get and at what point?
Okay, so I'm going to go through a task timing in a moment. I just thought I'd explain how I did the localizing first, and then we'll go to the trial. Okay. Okay, so this is one of the trials then in the localizer. So it's an example of the phonological localizer. So the subject sees a crosshair on the screen for 1,000 milliseconds, then they see the pair of words. And as soon as they see the pair of words, they're free to respond, plus they have an extra 1,700 milliseconds. On a third of the trials, they'll get TMS. And that will happen at 100, 200 and 300 milliseconds after the word appears. Okay, so these are just the results for the localizer then. So on the left, that's the semantic result, showing increases in reaction times with TMS versus no TMS. But of course this is no surprise, because this is what we were looking for. We were looking for areas where we'd find an increase with TMS, so that we could use that for the main experiment. And the same on the right then, for the phonological vein and pain. So I think we probably lost about 2, maybe 3 subjects where we actually just couldn't find a site. So we kept moving around. If we didn't find a site, we just had to lose them. So for the 4 subjects who did have MRI scans, they're the positions on the bottom right of where in circles the posterior site was and in crosses the anterior site. So we're pretty much on the border really between 45 and 47 for the anterior site, whereas we're within pars opercularis for the posterior site. Okay, so for the main experiment then, the subjects did 3 tasks. So there'd be a session for the anterior site and a session for the posterior site. The semantic task, they had to judge whether a pair of words meant the same thing, such as trip or journey. Some of these might not translate to American, but I think they probably do. The phonological task, then, they just judge whether a pair of words sounded the same or not. And then a control visual task, where they just had to decide whether 2 strings of letters were the same or not. And it should be something like, in this case, swapping 2 of the letters. Okay, so the timing is the same for the localizer tasks. But in this case, on 4 out of 10 trials, you get TMS at the same time as before, so 100, 200, and 300 milliseconds. Okay, so these are the overall results. So left panel semantic result, middle phonological, and on the right the control result. So in the top panel you can see there was no effect of TMS on accuracy. No significant effect. In the middle, we have the normalized reaction times. So because people's reaction times in general and with TMS are so variable, we decided to use a measure that was normalized. So we did a with TMS minus without over with by 100 to get our measure of the TMS effect for each individual. So you can see that the pattern is as follows. For semantic processing, you get an effect for the anterior site, but not the posterior site. Whereas for phonological processing, you get an effect at the posterior site, but not the anterior site. And for the control condition, there's no significant effect there between the TMS effects. So therefore, we concluded that you could, in fact, find double dissociation for semantic and phonological processing within this small region of Broca's area. Okay. I think I've all said that all before. Yep. Okay. So in this study, which came actually prior to the previous study, looking at semantic processing, Devlin et al. 2003 did also high-frequency TMS at 10 Hz. And they did repetitive TMS and also single-pulse TMS. And they were looking at semantic processing in anterior left inferior frontal gyrus. So they started off by doing an fMRI experiment comparing semantic, phonological, and perceptual processing. So the semantic task was to decide whether a word was man-made or not. The phonological task was just to decide whether the word had 2 syllables or not. And the perceptual was just to say whether the line above the word was shorter or not. And again, they had 300 milliseconds of TMS. And that's the timeline. Okay. Okay, so this is their overall fMRI result. So the one we're interested in for the TMS purposes on the left bottom in C. For the semantic greater than phonological decision, they did find this very small anterior activation that was greater for semantics than phonology. So they decided to stimulate this site to look for an effect. And in A and C, this is the result over the 300 milliseconds. So on the left top, you can see that with RTMS, there's a slowing of reaction times at that anterior site. but not without TMS. But for the perceptual task, there's no significant effect. So what the...
This is the 10 hertz 3 pulses, or this is the offline 1 hertz?
This is 10 hertz online 3 pulse. Okay, so over the 300 milliseconds, they get this pattern of effects. But of course, they still have this time window of 300 milliseconds. And in order to narrow that down, they decided to single pulses. So on different trials, they would do a single pulse at 100, 150, 250, etc. for the whole 300 milliseconds to see at what point the effect was actually happening. And you can see that in B, for the semantic task, it looks like the effect is really happening around 250 milliseconds, whereas in D, there was no significant difference at any of those time points versus no TMS. Okay. Yeah. So that's basically why I've shown this study, just an example of how you can refine the time window further. Okay. So TMS, then, we've seen, is a useful, non-invasive interference technique. It's spatially and temporally specific, depending on how you use it, of course. It has possibilities as a therapeutic tool, but I think this needs more work to see actually what's happening after you give TMS. And finally, it definitely adds greatly to understanding of areas that are involved in particular linguistic processes if used at high frequency.